Okay, then let's start. It's a biology stream. It's of biology class 7, chapter 13, photosynthesis in higher plants. Okay, let's start. There seems to be topics listed here. Let's see the topics. What do we know? Early experiment. Where does photosynthesis take place? How many pigments are involved in photosynthesis? What is light reaction? The electron transport chain. What are ATP and NADPH used? The C4 pathway, photorespiration, and factors affecting photosynthesis. These are all the topics listed here, almost 10 topics here. Okay, let's start. Let's start with introduction. All animals, including human beings, depend on plants for their food. Have you ever wondered from where plants get their food? Green plants, in fact, have to make or rather synthesize the food they need, and all other organisms depend on them for their needs. The green plant makes or rather synthesizes the food they need through photosynthesis and therefore are called autotrophs. You have already learned that autotrophic nutrition is found only in plants, and all other organisms that depend on green plants for food are heterotrophs. Green plants carry out photosynthesis, a physiochemical process by which they use light energy to drive the synthesis of organic compounds. Ultimately, all living forms on Earth depend on sunlight for energy. The use of energy from sunlight by plants during photosynthesis is the basis of life on Earth. Photosynthesis is important due to two reasons. It is a primary source of all food on Earth. It is also responsible for release of oxygen into the atmosphere by green plants. Have you ever thought what would happen if there were no oxygen to breathe? This chapter focuses on the structure of photosynthetic machinery and the various reactions that transform light energy into chemical energy. Okay, what they're saying here is this is all basic knowledge. We already know about autotrophs, basic knowledge from previous classes. What are autotrophs? Basically, any organism that can synthesize its own food is called as an autotroph. And here we are specifically calling plant an autotroph or green plant an autotroph because through its green pigment known as chloroplast, they can convert light energy into chemical energy and then into food. This is called photosynthesis, and this is done by green plants. And it's not solely restricted to plants. It can be done by it can be done by protozoans, fungi, etc., whoever have a pigment, a special pigment. And these are called autotrophs. And as for heterotrophs, all the organisms that directly or indirectly depend on the plants for as a food source, those are called heterotrophs. And they mentioned where does the plant get its energy? We know that plants are the source of all energy for other organisms. Then plant gets its energy from the sun. On earth, the basis of energy is basically sun. Without sun, there is no existence of life. And they also one more reason, they also mentioned two reasons why we need plants. One, they are the food source, primary food source. Two, they can liberate oxygen through photosynthesis. We know that plants use carbon dioxide, water, and their internal process to create food and oxygen. This oxygen will be liberated to the surroundings, and we know that oxygen is very essential for us humans and all organisms to survive. All aerobic process depend on oxygen. So obviously it's very, very important. Okay, that's with the introduction. Now let's come to what do we know. Let us try to find out what we already know about photosynthesis. Some simple experiments you may have done in earlier classes. Some simple experiments you may have done in earlier classes have shown that chlorophyll, a green pigment of the leaf, light, and CO2 are required for photosynthesis to occur. You may have carried out the experiment to look for starch formation. You may have carried out the experiment to look for starch formation in two leaves, a variegated leaf or a leaf that was partially covered with black paper and exposed to light. On testing leaves for presence of starch, it was clear that photosynthesis occurred only in green parts of the leaf present on in presence of sunlight. Okay, this is one experiment. What we know is, as I told, through chlorophyll, photosynthesis occurs, a green pigment of the plant through chlorophyll, through usage of light, CO2, and including water, which is very essential for photosynthesis. These all elements are required for photosynthesis, and they're converted into food. And one of the experiment we have carried out in our earlier classes is to find out where actually photosynthesis occurs. Photosynthesis occurs, we already know that it occurs in green parts of the plant, but how can we experimentally understand and prove it? Now what we take is two leaves, and one leaf we cover it partially with black paper. We covered it partially with black paper. Now we will expose them to sunlight for a few days, or for no matter how long, it depends. Now after exposure to sunlight, leaving for some time, we know that photosynthesis should occur and there should be food formation. And in plants, all the food is mostly stored in the form of starch. So if we test for starch, if there is presence of starch, then that means photosynthesis occurred. If no starch, no photosynthesis. So in the uncovered leaf, the complete leaf, it was seen that when we did the starch test experiment, the whole leaf was tested to have starch, meaning there was starch in every part of the leaf because it was a completely green leaf. Now, if we take a partially covered leaf and remove the cover and test for starch, then it was found out that the the starch was only present in the parts exposed to the sunlight. That meant sunlight was essential for photosynthesis. And another experiment to test out where it actually took place, green parts of the plant, that is taking a complete green leaf and a variegated leaf, basically with other colors etched into it. Now, in these two leaves, starch synthesis only occurred in green parts of the leaf. In other colored other areas, there was no starch synthesis. This also proved that this green pigment of the plant is also essential along with sunlight. Now let's go to CO2 or oxygen. Let's check it out. Another experiment you may have carried out, where a part of leaf is enclosed in a test tube containing some KOH soaked cotton which absorbs CO2, while the other half is exposed to air. The setup is then placed in light for some time. On testing for presence, the starch in the two parts of the leaf, you may have found that the exposed part of the leaf tested, tested positive for starch, while the portion that was in the tube tested negative. This shows that CO2 was required for photosynthesis. Okay. Basically, this is the experiment we saw for light, we saw for green parts of the plant. Now it comes to carbon dioxide. How we can prove it's also, or how we can know it's also essential for photosynthesis of plant? Then to understand this, we take two leaves. One, we just keep it outside. Two, we keep it in a test tube. And in that same test tube, we keep some KOH soaked cotton. KOH, potassium hydroxide. We take a cotton ball, and we soak it in KOH, that's potassium hydroxide. This, why we do this is potassium hydroxide, when we keep in a small container, it will react with all the carbon dioxide and convert it into something else. It basically absorbs carbon dioxide. It will reduce all the carbon dioxide in that present test tube or container to zero. Because of that, when we kept the leaf in the test tube and closed it with cotton balls soaked with KOH, all the carbon dioxide was reduced to zero. Now, if both the leaves, one in test tube, one outside, were exposed to sunlight, then and after some time were tested with star synthesis, we can find out that only the leaf that was outside exposed to atmosphere where carbon dioxide is there, only that one had starch presence, whereas the other leaf did not have starch at all. And because of this, we can conclude that carbon dioxide is essential for photosynthesis. Okay, that is the first topic completed. What are the first topics? Says photosynthesis in higher plants. What we know section. Three experiments were performed to find the necessity of carbon dioxide, light, and chlorophyll in plant in photosynthesis. How we did this? First experiment for chlorophyll. Take variegated leaf, comma, completely green leaf. Photosynthesis occurs only in green parts of the plant. Second, sunlight one, partially covered leaf. Photosynthesis occurs only in exposed part. Third, carbon dioxide leaf We take a leaf in test tube having KOH soaked cotton absorbs CO2 and no photosynthesis. Those are the essentiality experiments. Now let's come back. Let's see early experiments. Early experiments. It's interesting to learn about those simple experiments that led to gradual development in our understanding of photosynthesis. Joseph Priestley from 1733 to 1804 in 1770 performed a series of experiments that revealed the essential role of air in growth of green plants. Priestley you may call discovered oxygen in 1774. Priestley observed that a candle burning in a closed bell jar soon gets extinguished. Similarly, a mouse would soon suffocate in a closed space. He concluded that a burning candle or an animal that breathed the air both somehow damaged the air. But when he placed a mint plant in the small bell jar, he found that mouse stayed alive and the candle continued to burn. Priestley hypothesized as follows: plant restore to the air whatever breathing animals and burning candles remove. Can you imagine how would Priestley have conducted the experiment using a candle and a plant? Remember, he would need to rekindle the candle to test whether it burns after a few days. How many different ways can you think of light without disturbing the setup? Okay, let's see what Joseph Priestley did. Joseph Priestley, basically from 1933 to 1804, he was born in 1733. Okay, he conducted experiments in 1770. What experiments he conducted is basically the famous bell jar experiments. This is this whole dome-like structure is what we call a bell jar, and it completely isolates the system that is placed inside it. So, first, what he did was he took a bell jar and he kept a candle inside. First experiment, there was no mouse, only the candle was inside. After he kept the bell jar, then after some time, the candle got extinguished. Okay, that's first observation. Now, he kept a rat alone. He kept a single rat in a single bell jar. Then, after some time, the rat also suffocated and died. Then, that is the second observation. Third experiment, he kept both the rat and the candle together. This time, he observed that the mouse died or mouse suffocated and candle got extinguished faster than when they were placed alone. This is the third observation. Now, from these three observations, he concluded that a living candle, I mean a living mouse or a living thing, whatever it might be, and a candle seems to have damaged the air somehow inside the bell jar. That is what he concluded. He did not conclude that the air was completely gone or anything like that, because air was still there inside. There was no vacuum inside. He just thought the air was somehow damaged, and living beings could not survive in such damaged air. That's what we concluded. And let's go to the fourth experiment. Now, he took a rat, a candle, and it's very important to remember the name of the plant Joseph Priestley took. It is a mint plant. It's a mint plant. What Priestley took is a mint plant. He took this mint plant along with this rat and a candle. Now, what he observed was that after much time, after much more time than when he kept it at first, but this time neither did the mouse suffocate and die, nor did the candle get extinguished. And based on this single experiment, Joseph Priestley concluded that whatever the mouse and candle, a burning candle, were damaging the air, that was being restored or repaired by plants. That is what he concluded. So that was the experiment of Joseph Priestley. Now they asked some, can you imagine how he reignited the candle, kindled the candle, and all? It is not really required, but he might have, my guess, he might have used an automated kindler or something like that, using a similar setup as the one used by Priestley. Now let's go to the second experiment. This is also a similar setup as Joseph Priestley, but like basically a bell jar experiment. It's also a bell jar experiment, but by what Jan Ingenhouse did is he uses similar setup as one used by Priestley, but by placing it once in dark and once in sunlight. Jan Ingenhouse, 1730 to 1799, showed that sunlight is essential to plant process that somehow purifies the air fouled by burning candles or breathing animals. Ingenhouse, in an elegant experiment with an aquatic plant, showed that in bright sunlight, small bubbles were formed around the green parts, while the dark they did not. Later, he identified these bubbles to be oxygen. Hence, he showed that it is only the green part of the plant that could release oxygen. Okay, that's with the Jan Ingenhouse experiment. It's also very important to remember that Joseph Priestley was the one who discovered oxygen, even though he did not name it or anything like that. He was the first one, according to NCERT. Priestley discovered oxygen, and he used mint plant in his bell jar experiments. Now coming to Jan Ingenhouse, not much was given about him, only two paragraphs. He was born in also 1730, or just, anyways. Basically, what he did was he took this bell jar and he took both the mouse and also a burning candle. What he did was he kept this same exact setup once in sunlight and once in a dark place. We know that photosynthesis only occurs when there is presence of sunlight. So that meant that the oxygen was released only in sunlight, not in dark places. So, what did that mean? So, this experiment would be completely successful, like meaning the rat won't suffocate and the candle won't get extinguished if this setup was placed in sunlight. But if it was placed in the dark, this presence of this plant would not matter, because in the dark there is no release of oxygen. So when it's placed in the dark, there is no rays of oxygen, and the rat would suffocate, and the candle would get extinguished. That is the first part of this experiment. From this experiment, he concluded that the photosynthesis essentiality like requires light for photosynthesis to or oxygen to be released. Light must be there. That is the first conclusion. Now coming to second experiment, he took an aquatic plant. This aquatic plant, he took two different samples. One he kept in sunlight, and two he kept in dark places. And we know that whenever there is gas released in water, there will be bubble formation. Due to that, he showed that whenever in the plant which was kept in the sunlight, there was slight bubble formation in the leaves of the plant, on the leaves of the plant. And in the dark plant sample, one which was kept in the dark place, there was no bubble formation there. So from this experiment, he concluded that this oxygen was released only during when placed in sunlight. That is the gist of Jan engine house experiment. And Joseph Priestley used a mint plant to conduct his experiment, and he was the one who discovered oxygen. As for engine house, he was the one who named oxygen. He named it to be, later identified these bubbles to be oxygen. Then it was after he identified that bubbles to be oxygen, and showed that only green part of the plants could release oxygen. It was not until 1854 that Julius von Sachs provided evidence for production of glucose when plants grow. Glucose is usually stored as starch, as later studies show that green substance in plants, chlorophyll as we know of it, is located in spherical bodies, later called chloroplasts, within the plants. He found that the green parts in plants is where glucose is made, and that glucose is usually stored as starch. Okay, what they are saying is, this is now another, they did not give any kind of experiments here. It is just some kind of conclusions formed by Julius von Sachs. What Julius von Sachs at 1854 provided evidence for was that plants, when they grow, are producing glucose. Glucose is basically food. Glucose stored in plants. In plants, glucose is stored in form of starch. In animals, it is stored in form of glycogen. Two forms of storage of the same thing, that is glucose. And when plants are growing, they produce glucose and store it. Glucose is usually stored in the form of starch, and his later studies showed that green substance. Okay, first part is about the production of food, and this is in the form of glucose and stored in the form of starch. What is food? Glucose, and it's stored in the form of starch. Next, he understood like the green parts of the plant. Why the plant appears green is because there are special pigments known as chlorophyll present in circular spherical bodies in the plant cells, and these spherical bodies later are called as chloroplasts, which we know of. These chloroplasts contain the chlorophyll pigment. He also found that green parts in plants is where glucose is made. Glucose is made in the green parts of the plant, and that glucose is usually stored as starch. That's all for Julius von Sachs. Not any kind of experiment, but just some studies. Now consider the interesting experiment done by T.W. Angelman, 1843-1909. Using a prism, he split light into its spectral components and then illuminated a green alga, Cladophora. placed in a suspension of aerobic bacteria. The bacteria were used to detect the sites of oxygen evolution. He observed that the bacteria accumulated mainly in the region of blue and red light of the split spectrum. A first action spectrum of photosynthesis was thus described. It resembles roughly the absorption spectra by chlorophyll A and B. Okay, this is an experiment by T.W. Engelmann. He was born in 1843. What this scientist did is he took a prism. We know that the when we place when we take a prism, what is the function of a prism? It is to split light into its component, known as a splitting or resolution of light. This one, he did it. Now what he did is he placed this prism near a leaf, or basically a green part of a plant, and it directed the splitting components onto the leaf. And he also took some aerobic bacteria and kept them near the leaf. What is aerobic bacteria? Aerobic bacteria are bacteria which depend on oxygen, or basically which need oxygen for their metabolic pathways, or basically for their respiration. They need oxygen to produce energy. So they will get accumulated wherever oxygen is getting released. So when he passed this prism light, that is the components of light, onto the leaf, he observed that the bacteria got accumulated in the regions of blue and red. In the regions of blue and red light, where there are seven colors, that's with your violet, indigo, blue, yellow, green, orange, and red. In all those regions, the bacteria got accumulated most in the region of blue and red. This is known as an action spectrum. And he did not use a plant leaf, he used an algae. That algae is cladophora, just as how it is important to remember the mint plant used by Joseph Priestley. It's also important to remember cladophora used by E.W. Engelmann. Very, very important to remember. So basically, he understood that, and based on this experiment, he understood that the frequency of light, or basically the color region, was mainly focused in blue and red light. The main component that helped in photosynthesis was blue and red light, not any others. And it resembles the absorption spectra of the chlorophyll A and chlorophyll B. There is specific absorption spectrums of chlorophyll A and B. These are basically pigments. There are many kind of chlorophyll pigments, A, B, C, and D. We have seen or partly discussed them in plant kingdom. And this chlorophyll A and B absorb the frequency of blue color and red color light the most, which we will see in much depth later in this chapter about the frequency. I mean, about the wavelength of the light. And that is Engelmann's experiment. Now, by the middle of the 19th century, the key features of plant photosynthesis were known. Namely, the plants could use light energy to make carbohydrates from carbon dioxide and water. The empirical equation representing total process of photosynthesis for oxygen evolving organism was then understood as CO2 plus H2O in presence of light gives CH2O plus O2. An empirical equation is basically a basic equation, nothing more, nothing less. Now, by the middle of 19th century, meaning 1850s, By the time of 1850s, everyone understood what photosynthesis is. What is photosynthesis? It is basically the production of light, production of, I mean not production of light, production of food by green plants in presence of sunlight using carbon dioxide and water and liberating oxygen. This is the basic process of photosynthesis. Photosynthesis, and it's represented by this empirical equation. And CH2O is the empirical formula of glucose. We know glucose has a formula of C6H12O6. When we take 6 out, it will become CH2O. This is the empirical formula. That is what they wrote there. Where CH2O represents a carbohydrate. It can be a glucose, a 6 carbon sugar. A milestone contribution to the understanding of photosynthesis was then made by a microbiologist, Cornelius van Niel, 1897 to 1985, who, based on his studies of purple and green bacteria, demonstrated that photosynthesis is essentially a light-dependent reaction in which hydrogen from suitable oxidizable compound reduces carbon dioxide to carbon dioxide, I mean carbohydrates. This can be expressed as this one, okay, as before. Okay, let's explain this one. Okay, till the 1850s, people only knew that conversion or formation of food using these resources happens. They didn't know how it exactly happens, like it's going to, how carbon dioxide is going to get converted into, or how water is going to get converted into. And many people have a misconception that the oxygen released here, this oxygen, is from carbon dioxide. Whatever the oxygen and the carbon dioxide is getting converted into oxygen and getting released. But that is actually not the case. The oxygen released here is not from carbon dioxide but from water. Water will get reduced, or it will get electrolyzed into H2 and oxygen, O2, and O2 will be liberated. So basically, no, basically what I meant to say by that is nobody in the 1850 knew anything about the photosynthesis process. So when Cornelius van Niel, Cornelius van Niel, he discovered a part of the process and explained it, it was considered a major breakthrough or a contribution to our understanding of photosynthesis. So what this Cornelius van Niel said that okay, this is also very important. He performed experiments on purple and green bacteria. Joseph Priestley performed experiment on using mint plant. Angelman, his prism experiment, he performed using an alga, a green alga called cladophora. And here Cornelius van Niel, he performed experiment using purple and green bacteria. And there's one more, Jan Ingenhouse, he performed using an aquatic plant. There is no mention of the name of the aquatic plant, but it is just known that it is an aquatic plant. Now, Cornelius van Niel, who based on a study of purple and green bacteria, what he demonstrated that photosynthesis is a light dependent reaction that we all know. And in this light dependent reaction, hydrogen from suitable oxidizable compound reduces the carbon dioxide to carbohydrates. This is expressed by this equation. Okay, what he's saying is that when hydrogen from suitable oxidizable compound, oxidizable meaning it will lose electrons, so, or it will lose hydrogen, or yeah. Okay, what is oxidation and reduction? Just go through that. Oxidation is the removal of hydrogen or addition of oxygen. This is the basic definition. Oxidation is the addition of oxygen or removal of hydrogen. That is called oxidation. Oxidation, again once more, oxidation is the addition of oxygen or removal of hydrogen. Reduction is the addition of hydrogen and removal of oxygen. In these terms, we do not say electron loss and all. That does not matter here. According to biology here, what reduction and oxidation means oxidation either mean addition of oxygen and reduction of hydrogen, or it either means removal of reduction means it either means addition of hydrogen and removal of oxygen. That is what oxidation and reduction means. He described that Cornelius van Til described that a photosynthesis is a light dependent reaction in which hydrogen hydrogen from a suitable oxidizable compound. Why we call it as an oxidizable compound? Because we can remove hydrogen from it. From a easily oxidizable compound, we can remove hydrogen easily, and using this compound as a base, this compound will reduce the carbon dioxide. This easily oxidizable compound that can easily liberate hydrogen will come near carbon dioxide and reduces carbon dioxide to carbohydrates. Reduction meaning carbon dioxide will gain hydrogen and it will get converted into carbohydrates that can be expressed by this reaction. We already know that the components of carbohydrates is CH2O, meaning if we just add H2, that is hydrogen, meaning we reduce carbon dioxide, we can easily get our carbohydrates. So this oxidizable base which can add H2 to our carbohydrates is extremely mandatory, and this can be represented by this reaction. This 2H2A plus carbon dioxide in presence of light will give 2A, CH2O and H2. I believe A is not what A is not mentioned here, and according to what they mentioned above, it is basically just an oxidizable compound, meaning this A, like H2A, is basically an easily oxidizable compound which can easily liberate H plus, which can easily liberate H plus, not H plus, hydrogen, which can easily liberate or remove hydrogen from itself. It can also be water as well, as we do not take water here, as water can also act like that. When under light, it will liberate hydrogen and it will indirectly reduce carbon dioxide, and this hydrogen will attach to carbon dioxide. That is the formation of CH2O plus water is removed from there, and we will get 2A, that might just be oxygen. Yes, they mentioned here what this H2A is. It's basically water. In green plants, H2O is the hydrogen donor, which is acting as a oxidizable compound. Hydrogen donor and is oxidized to O2. Some organisms do not release O2 during photosynthesis, when H2 instead is the hydrogen donor. For purple and green sulfur bacteria, the oxidation product is sulfur or sulfate depending on the oxygen, I mean depending on the organism, and not O2. Hence he inferred that O2 evolved by green plants comes from H2O and not from carbon dioxide. This was later proved by using radioisotopic techniques. The correct equation that would represent the overall process of photosynthesis is therefore 6CO2 plus 12H2O in presence of light. 6 gives C6H12O6 plus 6H2O plus 6O2. Okay, what they mentioned here is from the above H2A, In most of the green plants, H2O is the oxidizable compound. H2O, like, it will easily give off like hydrogen, and it will reduce carbon dioxide into carbohydrates. So H2O is donating the oxygen. I mean, not oxygen, is donating H2, and it is getting liberated out as oxygen. So, but in case of green and sulfur, purple and green sulfur bacteria, these bacteria did not take H2O as their oxidizable compound, but instead preferred H2S, hydrogen sulfide. This is also having, this is also, there is no difference for them because this is also an oxidizable compound having H2. So their main need for H2 was to reduce carbon dioxide into carbohydrate. No matter was it from H2O or H2S, it didn't matter to them. So they quickly took the hydrogen from the source, that is the oxidizable compound, and liberated the product. In case of water, it was oxygen was the product, but in case of green and purple sulfur bacteria, the product, since they use H2S, was sulfur. And based on this observation, he concluded that the oxygen is liberated not from carbon dioxide but from water instead. And this was also later understood by radioisotopic techniques. And this is the correct equation for photosynthesis. It's also good to remember this one. 6 CO2 plus 12 H2O in presence of light gives C6H12O6 plus 6H2O plus 6O2. Only for this for this reaction, including 6 molecules of carbon dioxide, 12 molecules of water in presence of light, the only product, we only got a yield of 1 glucose molecule and 6 oxygen molecules along with 6 water molecules, where C6H12O6 represents glucose. The oxygen released is from water. This was proved using radioisotopic techniques. Note that this is not a single reaction but description of a multi-step process called photosynthesis. Can you explain why 12 molecules of water as a substrate are used in the equation given above? Okay, let's understand the equation. Basically, 6 carbon dioxide are there. We saw that glucose molecule has uh, 6 hydrogen, I mean 6 carbon molecule, carbon atoms, and these carbon atoms come from carbon dioxide, which has 6 carbon atoms. As for the 6 oxygen, they all come from carbon dioxide, and some are even released outside. Only, I mean, one oxygen atom from each carbon dioxide is released outside, and carbon will retain one oxygen atom, and hydrogen will attack this carbon, and it will form C6H12O6. And H12 comes from half of the hydrogen atoms present in this 12 molecules of H2O, and remaining half get converted to 6H2O, and the remaining oxygens, which lost hydrogens, will get converted into liberated oxygen. That's basically what is given here. Now that's the early experiments topic completed. Now let's write all the early experiments once more. Yes, let's start. Early experiment. First one is by Joseph Priestley. Bell jar experiment. Bell jar, comma, mouse, comma, candle, comma, mint plant, experiment. Conclusion. Concluded that damaged air damaged by candle and rat was restored by plants. First discovered oxygen. That's about Joseph Priestley. Second, Jan Ingenhouse. It might be very wrong spelling, but let's just know it's Jan Ingenhouse. Now, same bell jar mouse candle experiment. Bell jar in sunlight, mouse lived, bell jar in dark, mouse died. That is the experiment. I mean, not this is not the complete experiment. That is bell jar in sunlight, mouse lived. Okay, bell jar in dark, mouse died. And part B, aquatic plant experiment. Plant bubble formed in sunlight only. These are the two experiments by Jan Ingenhouse. Now let's go to third experiment. And this third experiment, this is not an experiment, but Julius, his name was Julius von Sachs. Experiment was not mentioned, but it is provided that he gave evidences that plants produce glucose as they grow Comma, that is one of the evidence he provided. Chlorophyll, green pigment, essential for photosynthesis, is in spherical bodies of plant cell called chloroplast. That's pretty much what he said. And also that one more is that he is like plant store. He also said plants store food, that is glucose, in the form of starch. The basis of our starch experiment we performed in lower classes depends on what Julius von Sykes said. So maybe if we didn't say that, we wouldn't have performed that experiment. Okay, next experiment by T.W. Angelman. Splitting of light using prism onto green alga, cladophora, and observed growth of anaerobic, not anaerobic, aerobic bacteria more in blue and red region of the light, where more photosynthesis, I mean more oxygen, was released. Hence, also more photosynthesis. That's with T.W. Angelman experiment. Now, next was Cornelius. His name was Cornelius. I think it was Cornelius Van Niel. Microbiologist Cornelius Van Niel studies on purple and green bacteria. Those Van Niel used green sulfur, purple green sulfur, used purple and green sulfur bacteria in his experiments. He said that he proved that photosynthesis releases or photosynthesis process itself, he explained photosynthesis is the process of oxidizable compound releasing hydrogen and therefore reducing carbon dioxide, or I'll just write CO2, reducing CO2 and thereby preparing carbohydrate. He showed that O2 released is from H2O and not carbon dioxide. If in most green plants H2O acts as the oxidizable compound, releases hydrogen and therefore the remaining oxygen is liberated. But in green purple bacteria, it was purple green sulfur bacteria, but H2S was acting as a replacement, and the liberated product was not oxygen but sulfur. What exactly, like sulfate, sulfur, or whatever it is, it's not, or sulfur dioxide, it's not clearly mentioned. It just said, yeah, compounds of sulfur basically. This is pretty much about, or the basic reaction of photosynthesis. Basic reaction he provided was H2A plus CO2 in presence of light, A plus CH2O plus H2. This is the basic reaction of photosynthesis. Now that's about the early experiment. Now I think we can go to the next topic. Okay. The third topic or the second topic, no, it's the third topic. Where does photosynthesis take place? Third topic seems to be a question. Where does the photosynthesis take place? Obviously, we can answer it as a green leaf or in the chloroplast, but let's see how. You would, of course, answer in the green plant or in the chloroplast based on what you earlier read in chapter 8. You are definitely right. Photosynthesis does take place in green leaves of plants, but it does so also in other green parts of the plant. Can you name some other parts where you think photosynthesis may occur? They're saying that it also occurs, it does occur in green leaf or in the chloroplast, but it doesn't only take place in the green leaf. It can also take place in other green parts of the plant. And this may be in the stem, in the petiole, where these are specific modification. In case of cactus, if we see, the leaves are modified into needles or basically thorns, what we say, due to reduce transpiration in the desert region. There's a special modification which we read in prior chapters. In case of Australian acacia plant, we see that the petiole of the leaf gets converted, it will just become fleshy and swell, and the leaf will fall off, but but the petiole will act as a photosynthesizing organ instead of the leaf. In case of openshea or cactus, it was the stem which was acting as a photosynthesizing organ, but in case of Australian acacia, it is the petiole of the plant. But normally leaves are the photosynthesizing organs, but here different modifications performed for different reasons, and there are special names for all of them. It is phylloclad and cladode respectively. Anyways, that is about this question. Can you name some other plants where photosynthesis may occur? For answering that question, I said this example, I told you these examples. Now let's continue. You would recollect from previous unit that mesophyll cells in leaves have a large number of chloroplasts. Usually the chloroplasts align themselves along the walls of the mesophyll cells such that they get the optimum quantity of incident light. When do you think chloroplasts will be aligned with their flat surfaces parallel to the wall? When would they be perpendicular to the incident light? Okay, we know in a dorsiventral leaf, basically a dicot leaf, there are two kind of mesophyll tissues, or basically that's not needed right now. Yes, let's just assume leaves have mesophyll tissue, something like this, and this mesophyll tissue, there is in this mesophyll tissue there is only one function, that is photosynthesis, and photosynthesis is through chloroplast. And if we take a single mesophyll cell, let's assume there is a mesophyll cell. In this mesophyll cell, there is a nucleus and there are chloroplasts, large number of chloroplasts. Okay, normally chloroplasts align themselves along the walls of the mesophyll cells, meaning they will align something like this. These are chloroplast cells. They'll align like this because in this way they can take all the incident light, they can absorb all the incident light to perfectly perform photosynthesis. Align to the walls. That is the thing they are saying. They will normally align near or parallel to the walls to get maximum amount of incident light. When do the chloroplast will be aligned with their flat surfaces parallel to the wall? And when would they be perpendicular to the incident light? Okay, it might be to reduce the incident light. I'm not completely sure, but we will not continue with that. Let's continue here itself. Now you have studied the structure of chloroplast in chapter 8. Within the chloroplast, there is membrane system consisting of grana, the stroma lamellae, and matrix stroma. There is a clear division of labor within the chloroplast. Okay, let's see the structure once more. We have already studied this in unit cell unit of life, but let's see it again. Diagrammatic representation of an electron micrograph of a section of a chloroplast. Basically, this is the outer membrane. This is the inner membrane, and there are connections here called stroma lamellae, and there are building like structures or coin arrangements called as grana, and the connections between these grana are called as stroma lamellae, and beyond those structures, the beside everything except grana, stroma lamellae, inner outer membrane, there is just stroma. Then there are ribosomes. In the stroma there are ribosomes. There is starch granules, not very important to remember. There is also lipid droplet. These two are not very important to remember, but ribosomes also can be neglected. But the main structure which help in photosynthesis are stroma lamellae, grana, and stroma. And we need to, it's also important to remember that uh, chloroplast is a double layered membrane consisting of outer and inner membranes. There are only few organelles which have a double membrane, including the nucleus and mitochondria. Like chloroplast is one of them. And chloroplast and mitochondria also have their own replicating system, meaning they have their own DNA and own ribosomes. And it is also important to remember that they have their own ribosomes, meaning it is independent of the cell. Cell has ribosomes, but chloroplast and mitochondria themselves have their own like ribosome, and they also have their own DNA. This is a specialty of mitochondria and chloroplast. There is an why story, but that will be discussed in the unit cell chapter. They replicate by themselves, and that is basically the structure of a chloroplast. And there is one more thing. Basically, if we have a coin like structure and we stack it on like this structure here, that is called a grana. But the single coin like structure is called a thylakoid. It's a thylakoid. A single layer is called a thylakoid, and the whole thing is called grana. That's about the structure of chloroplast. Now let's go. We have studied the structure in chapter 8. Within the chloroplast, there is a membranous system consisting of grana, the stroma lamellae, the matrix stroma. There is a clear division of labor within the chloroplast. This is also important. Remember, there might be a statement question. There is a clear division of labor within the chloroplast. The membrane system is responsible for trapping light energy and also for the synthesis of ATP and NADPH. What the membrane system, that is the outer and inner membrane system, does is its main function of the outer and inner membrane is to capture the sunlight or trap the light energy. Function is also very important. Remember, membrane system is responsible for trapping the light energy and also for synthesis of ATP and NADPH. These are energy molecules which we'll study later, more in later sections or in later chapters. In stroma, enzymatic reactions synthesize sugar, which in turns form starch. The former set of reactions, since they are directly driven, are called light reaction, photochemical reaction. The latter are not directly light driven but are dependent on products of light reactions, ATP and NADPH, hence do not distinguish, and hence to distinguish, they are called by convention called is dark reaction or carbon reactions. However, this should not be constructed to mean that they occur in darkness or they are not light dependent. Okay, what they are saying here is, first thing, after the basic introduction to chloroplast, they gave an information about how chloroplast membrane, which will trap sunlight, which main function of the membrane system is to trap sunlight, and it will convert this light energy into molecules, that is ATP and NADPH. These are energy molecules. ATP is adenosine triphosphate, and we'll see more about NADPH later. Now, after and this membrane system helps in the formation of this energy molecules. Okay, now coming to stroma, a kind of cytoplasm of this chloroplast. This stroma, it's this stroma undergoes enzymatic reactions to synthesize sugar, which in turn forms starch. Basically, the first they gave two information here. First thing, during the photosynthesis, like during the light is incident uh, on this chloroplast, at that time completely light dependent, this ATP and NADPH will be formed along with some enzymatic reactions which will lead to the formation of sugar, that is glucose. Glucose will be formed. Glucose will be synthesized, ATP and NADPH also will be synthesized. These three will be synthesized in the light reaction where they are directly completely utilizing the light. Now, after this, these enzymatic reactions which synthesize sugar and later forms starch. First is reaction to synthesize sugar, ATP and NADPH. This is done by many mechanisms, but this is directly dependent of light, meaning like without light it is impossible for those reactions to happen. It's not possible for those reactions to happen, and hence they are called as light reactions. The enzymatic reactions which lead to the formation of sugar or synthesis of sugar, then also the formation of ATP and NADPH. I'm not directly linking enzymatic reactions to formation of ATP and NADPH as they did not mention. The only enzymatic reaction mentioned here is synthesis of sugar, and there is also inclusion of ATP and NADPH synthesis, but it might not be an enzymatic reaction. All three are produced. This is the first reaction known as light reactions, which they do not occur in the absence of light. If we take a chloroplast cell, let's assume light is forming here. Now this will get converted into sugar. This is the first step of the reaction, and the second step of the reaction is sugar getting converted into glucose. I mean, not glucose. Glucose is sugar itself. Starch, sugar is getting converted into starch because that's how plant stores them. Now this is the first reaction. Now this is the second reaction. First is an enzymatic reaction now, and second, okay, first is a light reaction and second is called a dark reaction. But that, as NCERT said, it is not necessary that dark reaction needs to happen in the dark, as while light reaction needs to happen in the light, because Sugar can only be produced in the presence of light, but that is not the case for starch. It can be because sugar is already produced. The only thing the plant cell needs to do is convert this sugar into starch with necessary enzymes and chemicals or reagents. We can easily convert sugar into starch. There is no need for light. And even in light also, even if light was there also, there is no obstruction in the formation of sugar to starch. It does not depend on light. It rather than calling it that it only happens in the dark, we can say that the second reaction, that is the formation or conversion of sugar into starch, is an light independent reaction. But that does not mean I'm talking about the reaction itself. It does not need any kind of energy for this reaction to happen. It might need it, but it does not directly derive from the light, but some chemical energies. It is the reaction itself is light independent, but that does not mean it is completely independent of light, because sugar here is from light, a byproduct of or a product that is from the light reactions. Light reactions give sugar. Light reactions which are dependent on light, which only happen at the time of light, give sugar. And this sugar getting converted into starch is not necessarily dependent on light. Hence, it's called a dark reaction. But it will happen even in light also. It's just a second process, but just called dark reactions. Or even carbon reaction. That's about the third topic, which is very less if I have to say. They just told about chloroplast on cells. Okay, then structure of chloroplast, alignment of chloroplast. Information given here is alignment of chloroplast. Second, structure of chloroplast, how membrane system helps in synthesis of ATP and NADPH and traps sunlight. And about a brief, like not brief but like a basic idea of light and dark reactions. That's pretty much all. Let's write it. I think the topic name was, where does photosynthesis take place? Or we can change the color. Place of photosynthesis. Place of photosynthesis. Okay, now topic, first topic, alignment of chloroplast during photosynthesis, comma, chloroplast align themselves along the walls of the chloroplast, I mean of the cell, to maximize incident light. Diagram representation. Let's take a square cell, which is how normally plant cells are. That became a rectangle, but let's draw it again. Okay, let's assume this is a cell. It might be better to draw chloroplasts in green, but I'm going to do it anyway. I'm not going to do it. This is alignment to maximize their sunlight harvesting ability. Now, structure of chloroplast. Let's draw a diagram, and this will be in green. That's about the main structure. Now, they did mention something about it happening specifically in stroma. Okay. The membrane system traps sunlight and forms ATP and NADPH. In stroma, synthesizes sugar via an enzymatic reaction. Synthesizes sugar via an enzymatic reaction. Light reaction. Sugar gets converted to starch, also in stroma. Dark reaction. Dark reaction is independent is dependent on light reaction products, but independent of light itself. Indirectly dependent, but it mainly depends on light reaction products. Light, not production, light reaction. Okay, that's about dark reaction. That's pretty much all about this topic. Now let's go to next topic. Okay, the fourth topic is about how many types of pigments are involved in photosynthesis. But before that, let's take some break. There was a much longer break than anticipated, but let's continue with the fourth topic. How many types of pigment are involved in photosynthesis? This is also a relatively small topic. So let's see. Looking at plants, have you ever wondered why and how there are so many shades of green in their leaves, even in the same plant? We can look for an answer to this question by trying to separate the leaf pigments of any green plant through paper chromatography. A chromatographic separation of leaf pigments A chromatographic separation of leaf pigments shows that color that we see in leaves is not due to single pigment but due to four pigments, that is chlorophyll A, bright blue to green in chromatogram, chlorophyll B, yellow and green, xanthophylls, yellow, carotenoids, yellow to yellow orange. Okay, we see number of colors, number of shades of green in plants. Why we see them is due to pigments. And you can identify these leaf pigments through an experiment called as paper chromatography. And through this experiment, you can identify what are the different kind of colors or pigments we see in plants. And the most common pigments are chlorophyll A, which has a bright uh, blue-green color. These color combinations are important to remember. Chlorophyll A, blue-green, chlorophyll B, yellow-green, xanthophylls, yellow, carotenoids, yellow to yellow-orange. Carotenoids, carrots. Carrot is orange, carotenoids are orange. Xanthophylls are yellow in color. Chlorophyll B is yellow green. Both chlorophyll A and chlorophyll B are somewhat greenish, but they have a mix of blue green and yellow green. Now xanthophylls are solely yellow, carotenoids are yellow to yellow orange. Now let us now see the role of various pigments play in photosynthesis. Pigments are substances that have an ability to absorb light at specific wavelengths. That is the main use of pigments. Why only pigments can absorb specific like light at specific wavelength is because light, if we see the spectrum of light is basically an electromagnetic spectrum which has many wavelengths, many frequencies, etc. And pigments which have an ability to show a specific color, when the light hits a surface, it completely reflects black or completely gets absorbed. The reflected light will have every if it is completely reflected, it will have the same colors or same frequency as before incident and after reflection. But in case some special compounds which can absorb specific sequences of the light. For example, if this the reason why plants look green in color is because chlorophyll is a special pigment which can absorb, let's just say, the frequency other than green color. Every like frequency of light which is other than green, every other color, like mostly red, I mean blue and red, which we saw from I think Engelmann's experiment, except green color. Ironically, the the reason, normally when people think, it might be intuitive to say green is the color they absorb, but it is inverse. Green is the only color that is mostly not absorbed and hence reflected. Because of that, we perceive plants to be green in color. Pigments are substances that have an ability to absorb light at specific wavelengths. Can you guess which is the most abundant plant pigment in the world? Let us study. It's most likely chlorophyll A. Let's confirm. Most abundant plant pigment in the world. The most abundant plant pigment in the world is, as I guessed before, chlorophyll A. Most abundant plant pigment in the world is chlorophyll A. Let us study the graph showing. Okay, let's see the graphs. The first graph is graph showing absorption spectrum of chlorophyll A, B, and the carotenoids. Absorption spectrum, like which will absorb more amount of light. Absorption of light by chloroplast pigments. If we see at a specific sequence, chlorophyll, this blue color is chlorophyll A, and if we see, this is absorption depending on light frequency. This is frequency, x-axis is not frequency, wavelength. At certain wavelength, the chlorophyll A has reached its peak. Similarly, chlorophyll B as well, in this frequency it had reached its peak. And for carotenoids as well, these are the peaks where relatively if we see chlorophyll A has the highest absorption frequency, I mean absorption of light, or relatively high absorption of light, because it has two peaks. Although this peak is smaller than chlorophyll B, here there's another peak covering up for it, which is higher than chlorophyll B's peak. Now that's about the spectrum, but there might be, it's important to remember the graph, because they might give the graph and ask which are carotenoids, or identify chlorophyll B, chlorophyll A from the graph. And as there will be no colors in the graph, we can't identify based on color. We need to know specific peaks which and where they reach the peak. The first and last peaks are chlorophyll A. The second and the last second peaks are chlorophyll B. The two simultaneous peaks are carotenoids. And basically these are the only three pigments here. Now, a rate of photosynthesis measured by oxygen release. Graph showing action of spectrum photosynthesis. They all are at same wavelengths here, 400, 500, 600, 700. And where there are peaks, the oxygen release is relatively higher, where here there is completely like no peak here. There also the photosynthesis reduced. This is at specific frequencies of light. These two peaks are the reason why in Angelman's experiment it showed that plants, like the aerobic bacteria, got accumulated where it was the region of blue and red light, and instead of other region. Blue, if we see, has a relatively high frequency and low wavelength, meaning it should be near this, that is why there is a peak here. And here red has the highest wavelength among the visible range, that is the end of the spectrum, almost here. At both areas there is a peak here. Now photosynthesis, where there are higher peaks here in this, there's also the higher photosynthesis rate. Higher the light absorbed, higher the rate of photosynthesis. Rate of photosynthesis, absorption. Okay, graph showing action spectrum of photosynthesis superimposed on absorption spectrum of chlorophyll. Okay, basically this one, chlorophyll A. We saw two different graphs here, this one and this one. And when we mix both these graphs, or when we superimpose both these graphs, we will get graph C. And we only considered chlorophyll A here, not other pigments. We considered only chlorophyll A. So whenever there is a peak here, there is a photosynthesis also a peak, and when this reduced, this also reduced, and here there's another peak. That's pretty much all about the graphs. Let us study the let's read. Let us study the graph showing the ability of chlorophyll A pigment to absorb lights of different wavelengths. Of course, you are familiar with wavelength of the visible spectrum of light known as Vibgyor, violet, indigo, blue, green, yellow, orange, red. From figure 13.3a, can you determine the wavelength color of light at which chlorophyll A shows maximum absorption? And the first peak where it is basically blue light. Chlorophyll absorbs the most of the blue light, shows the maximum absorption. Does it show another absorption peak? Yes, it shows at red the maximum wavelength also. At like if we see this is around 400 to 430 nanometer. Here it is, I believe 670 nanometer at these two regions. Absorption peak at any other wavelengths too? If yes, which one at red? Now look at figure 13.b. It's very important to understand the Angelman's experiment before going to this. Angelman's experiment is basically the one we saw in early experiment about the prism splitting light one. Now look at, we have gone through it. Now look at the 13.3b showing the wavelengths at which maximum photosynthesis occurs in a plant. Can you see that the wavelengths at which there is maximum absorption by chlorophyll A, that is the blue and the red regions, also show higher rate of photosynthesis. Hence, we conclude that chlorophyll A is the chief pigment associated with photosynthesis. But by looking at figure 13.3, can you say that there is complete one-to-one overlap between absorption spectrum of chlorophyll and the action of the spectrum of photosynthesis? So basically, based on this, the higher the peak of chlorophyll A, the rate of photosynthesis is also significantly increasing. Like if we compare, there's a peak of carotenoids here slightly after chlorophyll B, and there's a very high peak of chlorophyll B slightly after chlorophyll A. Even though these two peaks, there are vast absorption here, but still if we see the maximum uh, photosynthesis rate was seen to be not near those peaks but chlorophyll A. Even though the absorption of light was slightly less compared to both these combined, the photosynthesis rate was maximum close to the peak of chlorophyll A. And if we see the second peak of chlorophyll A, this is the chlorophyll A, there is a exponential increase of photosynthesis rate here as well. So based on this, we can conclude that chlorophyll A is the chief pigment of the plant which helps in photosynthesis. But even though it is a chief pigment, if we see this is the mixture superimposed of the both graph, if we see even though absorption of chlorophyll A is completely stopped at near 500 nanometer wavelength, in this case here, the photosynthesis, although declining, did not completely stop. There is not a complete overlap. This is not a complete overlap. That basically meant there are influences of the carotenoids, chlorophyll B as well. It's not solely just because chlorophyll A is the chief pigment, it means that it has higher efficiency and just efficiency. Because of that, it was named the chief pigment, but that does not mean there's a complete overlap. Even the other pigments also help in the process. That's what we can understand when we say there is not a complete overlap between rate of photosynthesis and absorption of the absorption spectrum of the like chlorophyll. These graphs together show that most of the photosynthesis take place in the blue and red regions of the spectrum. Some photosynthesis does takes place at the other wavelength of the visible spectrum. Let us see how this happens. Though chlorophyll is the major pigment responsible for trapping light, The other thylakoid pigments like the chlorophyll B, xanthophylls, carotenoids, which are called accessory pigments. Important to remember the word accessory pigments and also the names of the accessory pigments: xanthophylls, carotenoids, chlorophyll B, and chlorophyll C. These are all called as accessory pigments. They also help also absorb light and transfer the energy to chlorophyll A. This is also very important to remember. They do not have direct action. They do capture the sunlight and transfer it to the pigment chlorophyll A. Transfer it to the pigment chlorophyll A. They do not directly take action. Only their function is transfer energy, trap energy, transfer to chlorophyll A. Indeed, they not only enable a wider range of wavelength incoming light to be utilized for photosynthesis, but also protect chlorophyll A from photooxidation. Also very important use of the accessory pigments. Two functions of the accessory pigments: they can uh, they can trap light energy from various different wavelengths. That is the first use, and send it to chlorophyll A. Second use, they can protect the chlorophyll A pigment from overexposing itself and undergo photooxidation. This is kind of a degradation of the pigment chlorophyll A, which if happens, it will not be able to function very well. Now that's it for this topic. Let's write it. Pigments of plants. Accessory pigments. First slide left, let's write chief pigment. Chlorophyll A, blue green, bright. Accessory pigments. Chlorophyll B, yellow green, xanthophyll, yellow, and carotenoids, orange, yellow. Now those are the accessory pigments. Now functions of accessory pigments. One, capture a larger variety of light wavelengths. Transfer to chlorophyll A. To protect chlorophyll A from photooxidation or degradation. That is main topics, I mean main points of this topic, including the graphs. Anyways, now this is but what is a light reaction. This is going to be the start of the main topics, the main actual reactions. Okay, let's start with the topic. Topic 5. What is light reaction? What is light reaction? Light reactions or photochemical phase include light absorption, water splitting, oxygen release, the formation of high energy chemical intermediates, that is ATP and NADPH. Several protein complexes are involved in this process. The pigments are organized into two discrete photochemical light harvesting complexes within the photosystem 1, that is PS1, and photosystem 2, PS2. These are named in the sequence of their discovery and not in the sequence in which they function during the light reaction. The LHC are made up of hundreds of pigment molecules bound to proteins. Each photosystem has all pigments except one molecule of chlorophyll A, forming a light harvesting system called antenna. These pigments help to make photosynthesis more efficient by absorbing different wavelengths of light. The single chlorophyll A molecule forms the reaction center. The reaction center is different in both the photosystems. In PS1, the reaction center chlorophyll A has an absorption peak at 700 nanometer and this is called P700, while in PS2 it has an absorption maxima at 680 nm, that is nanometer, and it's called P680. Okay, it's a small topic, but let's see. The light reactions, we saw that light reactions include the formation of sugar in stroma, the enzymatic reactions which lead to the formation of sugar in the stroma. Second, it's the membrane synthesizing ATP and NADPH. There is also a light reaction. These two are light reactions. They are also called photochemical reactions or photochemical phase. These include light absorption or the main energy source, that is the light absorption. Next, they split the water molecules. We saw that according to Cornelius Van Neel, I believe, he said that the splitting of water, basically it is acting as an oxidizable base by giving off hydrogen, and this hydrogen will attack carbon dioxide and form carbohydrates. Splitting of hydrogen or water is basically this process. We are oxidizing water to give out hydrogen. Then oxygen release, that is very closely related to water splitting, and the formation of high energy chemical intermediates, that is ATP and NADPH. These what ATP and NADPH are, they are high energy. They have high energy by breaking the chemical, like these are intermediates. Why they are called intermediates is because they are their main function, they have energy in their bonds. When they release their energy, like when their bonds are broken, they will release certain amount of energy that helps in doing work, and that is why they are called chemical intermediates. They do not, their purpose is not to exist forever. Their main purpose is to get converted into less energy molecules giving off energy. They're kind of a temporary permanent storage of energy. It's not completely permanent, it's a temporary storage of energy. They are ATP and NADPH. These are high energy chemical intermediates. Several protein complexes are involved in this process. There is a process of photosynthesis or the photochemical phase of the photosynthesis. The pigment, several protein complexes are involved in this. The pigments are organized. Basically, pigments are the ones which trap and collect the sunlight. So uh, these are aligned into several complexes, and two complexes organized into two discrete photochemical light harvesting complexes. This is a light harvesting complex, also referred to as LHC. This is a harvesting complex, and there are two kinds of light harvesting complex. The pigments are organized into two discrete photochemical light harvesting complexes within the photosystem 1 and photosystem 2. Why they are named photosystem 1 and photosystem 2 is due to photosystem 1 was discovered before photosystem 2, not because photosystem 1 is going to function before photosystem 2. No, it is only because photosystem 1 was discovered prior to photosystem 2 was a naming sense made. The pigments are organized into two discrete photochemical light harvesting complexes. Okay, there are the classification is something like this: light absorption done by two different systems, that is photosystem 1 and photosystem 2, that is PS2. And in these photosystems there are light harvesting complexes. In I'm not exactly sure in PS1 there are specific limitations, but this is just for example as what the system is based on. Light absorption has two different photosystems. It's based on two different photosystem, and each photosystem has two different kinds of light harvesting complexes. Wait, no. Each have different one. Okay. Okay. In photosystem 1, the light harvesting complexes has a peak of 700 nanometer. Hence, it is called P700. Whereas in photosystem 2, the peak of the light harvesting complex is at 680 nanometers. Hence, it is called P680. These are light harvesting complex reaction centers. Okay, let's get back to the classification once more. Light absorption is done through two discrete photosystems, and photosystem 1 has light harvesting complexes within which there is a chlorophyll A molecule that has a maxima of 700 nanometer absorption rate in absorption spectrum, whereas there is in PS2 system there is also light harvesting complexes there, but there the peak of the light harvesting system like reaction center is only 680 nanometers, so hence it's called P680. And this absorption spectrum is about the chlorophyll A molecule only. Okay, let's read this again to understand. Light reaction for photochemical phase include light absorption, water splitting, oxygen release, and the formation of high energy chemical intermediates ATP and NADPH. Several protein complexes are involved in this process. The pigments are organized into two discrete photochemical light harvesting complexes, which the photosystem 1 and photosystem 2 These are named in sequence of their discovery and not in the sequence in which they are function during the light reaction. The light harvesting complex are made up of hundreds of pigment. Okay, we are coming to the structure of the light harvesting complexes. The light harvesting complexes are made up of hundreds of pigment molecules bound to proteins. Each photosystem has all pigments except one molecule of chlorophyll A, forming a light harvesting system called antenna. Basically, what is a light harvesting complex? Is basically hundreds of pigments together. There will only be one chlorophyll molecule in a single light harvesting complex, but there will be hundreds of other kind of pigments in each light harvesting complex. If we take this is a light harvesting complex, the light harvesting complex, these are all pigment molecules. In the center of all these pigment molecules there will be a reaction center. This is the reaction center here. This is the chlorophyll A molecule, and this is a photon, basically light. Photon will or light will fall on these other all kind of other pigments, and the energy will be transferred like this. We saw before that auxiliary pigments do not directly indulge in the process of chemical reactions. Their main aim is to absorb varied kind of light, light wavelengths and send it to chlorophyll A. That is their main function. So that is what they do. There is a cluster of pigments here with a single core, that is the chlorophyll A molecule. The light will fall on all the other pigments and it will get transferred to the chlorophyll A. That is the chain shown here. This is also called a reaction center, and this whole thing is called, this whole complex is called as an antenna. And this reaction center will send the energy to the primary acceptor, which we'll see later, more about later. The pigments help make the photosynthesis more efficient by absorbing different wavelength of light. The single chlorophyll molecule forms the reaction center. The reaction center is different in both photosystems. The PS1 reaction center has an absorption of 700, that is P700. PS2 has P680. Okay, they showed something as a primary acceptor here. Let's see how actually the reaction center will send energy to the primary acceptor. What will be the form reaction center core like P680 in PS2 or P700 in PS1 has a pair of specialized chlorophyll A molecules. When these absorb energy from photons directly or form auxiliary pigments via resonance transfer, the energy excites their electrons. Okay, All the energy will be concentrated in electrons. The electrons will go into high energy states. Basically, they will go into higher orbitals. They will get excited, high energy electrons. So the absorbed light energy is converted into excited high energy electrons. What will the reaction center send to the primary acceptor? The excited electrons from the chlorophyll A molecules are transferred to nearby molecules called the primary electron acceptor. This means the reaction center sends out high energy electrons, not photons, not ATP directly. Now, what is a primary acceptor? The primary acceptor is the first electron carrier in the photosynthesis. Photosynthetic electron transport chain. It sits right next to the reaction center and is specialized to trap the excited electron so it does not fall back into the ground state. Let's not care about the primary acceptor pheophytin or whatever it is. It does not involve our level. Are the electrons are the energy from the other pigments also sent to chlorophyll A through excited electrons? The energy from auxiliary pigments like chlorophyll A, what happens is how auxiliary pigment works. Auxiliary pigment absorb light at wavelengths chlorophyll A cannot absorb efficiently. The energy they absorb excites their own electron to high energy states, but these electrons don't leave the pigments. Instead, the excited energy is transferred resonance wise, like a vibration or handover of energy, not electrons, from one pigment to reaction center. Only the special pair of chlorophyll A molecules in the reaction center actually donate an excited electron in the primary acceptor. Is there will chlorophyll A molecule in the entire most of them are antenna pigments whose job is to absorb light and pass energy towards the reaction center. Reaction center is inside the photosystem. The special pair of chlorophyll A form the reaction center. They are the only ones that there are many chlorophyll A pigments. Many chlorophyll A molecules are present in the antenna complex along with chlorophyll B and carotenoids, but only one special pair, two chlorophyll A molecules per reaction center, actually perform the charge separation. There are a pair, not single one, two chlorophyll A molecules which get excited electrons. How can the pigments Whenever the reaction center loses an electron, the special pair P680 or P700 gets oxidized, will become P680, P700. One more definition of oxidation is losing of electrons is called oxidation and gaining of electrons is called reduction. This oxidized chlorophyll is very strong oxidizing agent, meaning it can undergo reduction easily. It pulls in an electron from somewhere else, wherever it might be. And where does this electron somewhere else come from? Where do the replacement electron come from? In photosystem 2, electrons are replaced by splitting water molecules, that is photolysis at oxidation level, where reaction is something like this, where electrons will be donated by hydrogen. Basically, hydrogen will lose all its electrons, become H plus, and there will be 4 electrons and O2. So P680 takes its missing electron from water, which is why PS2 is the ultimate electron donor in plants. Meaning it can, why it is the ultimate electron donor in plants must most likely is because there is a high abundance of or basically a necessity for water molecules to be split like this. Due to reduction of carbohydrates or basically carbon dioxide into carbohydrates, there is a necessity for water to split. So this is actually a necessity through which PS2 gains. As PS2 can take electrons from a necessary process, the electrons don't get wasted and it can easily replenish its electron, and as such it can donate more as well. In photosystem 1, if we see electrons lost by P700 go to the primary acceptor and go down the electron transport chain, and replacement electrons come from plastocyanin, which carries electron from PS2. Okay, so basically it is also a source from electrons of water splitting. The electron flow, basically water is the main, water splitting gives most of the electron. Water to PS2 to electron transport chain to PS1 to NADP plus, which will get reduced into NADPH. And if this one loses electrons, it will get from PS1. If this PS1 loses electrons, it will get from electron transport chain. If electron transport, like what electrons the electron transport chain will transfer, it is from PS2. Where does PS2 get its electron? From water splitting. Finally, the source of all electrons in plants is through water. If it was any other kind of, like in, we saw in purple green bacteria, I mean purple green sulfur bacteria, we saw there was H2S instead of like hydrogen sulfide instead of water. In that case, it will get electrons from hydrogen sulfide itself. Chlorophyll molecules themselves never get used up, they just go through a cycle through excitation, electron loss, and replenishment. Reaction center doesn't get exhausted because P680 is refilled with electron from water. Okay, let's go back then. And this diagram is also very important to remember. Every label of this diagram is important to remember. It's not that hard to remember either. That's about the light harvesting complex, but let's write it down. Or maybe it's better not to write it as light harvesting complex. Or let's just write it for now. Group of pigment molecules transfer energy to chlorophyll A reaction center. Chlorophyll A excited electrons go to primary acceptor. This is the main LHC mechanism to photosystem. PS1 and PS2. PS1 reaction center has 700 nanometer absorption spectra. PS2 only has 680 nanometer. P700 nanometer being 10 power minus 9 meter. P680, okay. That's all the information given in this and including the diagram. If I had to write the diagram, it will be something like this. Reaction center, primary acceptor. That's a lighting effect. Auxiliary pigments. Reaction center. Energy transfer. That's the diagram of LHC. Okay, now let's go back. The electron transport chain. Sixth topic of this lesson. We are not even halfway through the topic or chapter. Okay, the electron transport chain. In photosystem 2, the reaction center chlorophyll A absorbs 680 nanometer wavelength of red light, causing electrons to become excited and jump into an orbit farther from, wait a second, I think I forgot to write something from our prior topic. Okay. PS1 sends electron excited to primary acceptor. PS1 replenishes lost electron from PS2, and PS2 replenishes electron from water splitting, called photolysis. Then that's for the topic. Okay, I think we have completed that topic information. Let's go back. In photosystem 2, the reaction center chlorophyll A absorbs 680 nanometer wavelength of red light, causing electrons to become excited and jump into an orbit further than the atomic nucleus. Okay, that's what we read. But this is similar to how photosystem 1 works as well. Here as well, the electrons jump from normal to ground to excited state and then went to primary acceptor. Here as well, in photosystem 2 as well, the reaction center, but here it's a P680 nanometer reaction center. Here as well, the light will cause, this is the red light. 680 nanometer is a red light spectrum, like it's the range of red light, causing electrons to become excited and jump into an orbit farther from the atomic nucleus. Basically, they are leaving the atom. These electrons are picked up by an electron acceptor, which passes them to an electron chain. In case of PS1, there was the primary acceptor. Here, I'm not sure what they call it, but here they call it just as an acceptor, an electron acceptor. Basically, light on PS2 gives electron to acceptor to transport chain, ETC, electron transport system of consisting of cytochromes. I'm not sure what cytochromes are, but cyto means cell, chromes means color, some kind of colored cells. And it might be important to remember of the name cytochromes. The electron systems, the electron transport system or electron transport chain consisting of cytochromes. This movement of electrons is downhill, meaning from higher potential to lower potential, in terms of basically they are exhausting their potential, going through this electron chain. They're losing energy. In terms of oxidation reduction or redox potential scale, the electrons are not used up as they pass through the electron transport chain, but are passed on the pigments photosynthesis PS1. Simultaneously, electrons in the reaction center of PS1 are also excited when they receive red light of wavelength 700 nanometer and are transferred to another acceptor molecule that has greater redox potential. Okay, basically light falls on LHC, electron acceptor of photosystem 2, which will send to electron transport system. This is the chain we saw before, where LHC replenished its electron from the electron transport chain from this acceptor to this is the photosystem 2 acceptor. And all the electrons accumulated in the photosystem 1 will go to the electron acceptor. This is a primary electron acceptor in photosystem 1, which will convert NADP plus into NADPH. Basically, it will reduce NADPH. There's a reduction reaction going on. These electrons then are moved downhill again, this time to a molecule of energy rich NADP plus. The addition of these electrons reduces NADP plus to NADPH plus H plus. This whole scheme of transfer of electrons starting from PS2 uphill to the acceptor, down the electron transport chain to PS1, excitation of electrons, transfer to another acceptor, and finally downhill to NADP plus, reducing to NADPH plus H plus, is called the Z scheme, probably because it looks like a Z. If we see here, Z, inverted Z. It looks more like an N to me, but let's go with the Z scheme. Due to its characteristics, shape, this shape is formed when all the carriers are placed in a sequence on a redox potential scale. That's about the electron transport system. Okay, the information given here is after the PS2, here PS2 is there, light falls on the LHC of the the second photosystem. Here, after it falls, electrons in the chlorophyll A, that is a P680, the reaction center, get excited. All the energy is transferred to the PS, like the reaction center of the light harvesting complex, and the electrons in the reaction center get excited. These excited electrons are transported to an electron acceptor of the photosystem 2. Then this electron acceptor will transfer them to an electron transport system. This system is consisting of cytochromes, and here the movement of electron is downhill. This is in terms of redox potential. I'll check that out later. Just let's just understand it does not require energy. Like what this downhill means is that there is no extra special energy required to transport the electrons. They are basically going on their own from high potential to low potential. Electrons do not lose their energy, nor are they used up in this transport system. And if we see, they're not mention anything here, but if we see closely here, they mention it. They mention that when it is going through the electron transport chain, ADP plus IP gives ATP. This is synthesis of ATP molecule is also happening here. Along with NADPH, ATP molecule is also getting synthesized here. But I'm not sure why they did not mention it here. Okay, that's about that. Now, after the electron transport chain takes these electrons to the reaction center, that is P700 reaction center. Here the electrons get excited through the energy collected by LHC1 and get sent to the primary electron acceptor, and this will give the electrons, basically reduce NADP plus. And this whole scheme is called as a Z scheme. Most important thing is this diagram, where it also serves as a diagram question and also easy to understand and remember through the diagram than the text. Let's draw the diagram ourselves. This is the electron transport. Electron transport. Electron transport system. Made of consisting of cytochromes, makes downhill movement of excited electrons received from the light harvesting complex possible and retain their energy. This is the most of the important paragraph except the Z scheme. Let's draw the Z scheme. Sun when we want an oval, we don't get an oval. This is the first LHC. This will get excited from the sun's light, and this is the acceptor. Electron acceptor. Now here it will go to the electron transport chain. Let's use a different color. Transport chain to second LHC. This is PS2. Now it's again sent to another primary acceptor. PS1. Both seems to be the same color anyways. NADP plus will become NADPH plus H plus, and there is another reaction here. ADP plus PI become ATP. This is the whole Z scheme. Z scheme, very important number, the name as well as the diagram. Now, any missing information? I think that might be all. Okay, let's go back. Now coming to splitting of water. This is a sixth topic. Okay, this is a subtopic under the electron transport topic. Subtopic 1. You would then ask, how does PS2 supply electrons continuously? The electrons that were removed from photosystem 2 must be replaced. This is achieved by electrons available due to splitting of water. The splitting of water is associated with PS2, water split into 2H plus nascent oxygen and electrons. This creates oxygen or the net products of photosynthesis. The electrons needed to replace those removed from the photosystem 1 are provided by the photosystem 2. H2O gives 4H plus O2 plus 4 electrons. We need to emphasize here that water splitting complex is associated with PS2, which itself is located in the inner side of the membrane of the thylakoid. Then where are the protons and oxygen formed likely to be released, in the lumen or on the outer side of the measure? Okay, this one I have already mentioned or explained this, how like why we need more electrons, like how it will be provided. It's through water splitting, and this is the reaction which we saw before itself. Now coming to the, like, where the water splitting complex is associated. Water splitting complex is basically, as the name suggests, the complex which helps in the splitting of the water. It is associated with PS2. As we saw before, PS2 is the one that supplies electrons, a major electron donor of the chloroplast, where it supplies electrons to both PS1 and other also in electron chain also. That means that only like PS2 is the most likely one to be associated with water, as they have said. And this water splitting complex is actually present, like associated with PS2, and PS2 itself is actually present inside the, okay, PS2. There are two different systems, PS1 and PS2. PS2 has 680 nanometer reaction center. PS1 has 700 nanometer reaction center. Now this PS2 is physically located inside the membrane of thylakoid. If there is a thylakoid membrane, then inside the thylakoid of the thylakoid membrane there is PS2. If it itself, like this PS2 itself is located in the inside membrane of the thylakoid, then where are the protons H plus here formed in this reaction and oxygen likely released? Lumen meaning inside the thylakoid space, inside the thylakoid membrane, or outside the membrane. I'm not sure. Let's ask. The oxygen evolving complex, also called the water splitting complex, is part of photosystem 2. It's located in the inner lumen facing the thylakoid membrane. Where the product released? Proton released directly in the thylakoid lumen, contributes to proton gradients. Okay, this is very important, as for later we will see more about proton gradients inside the lumen versus low H plus in stroma. This gradient later derives, yeah, this is what I was talking about, like ATP synthesis through through ATP synthase. Electrons immediately transfer to P680, the oxidized reaction center of PS2. Oxygen released as a byproduct. Since electron chain faces the lumen, O2 is first produced in the luminal space, but O2 is small and non-polar, so it diffuses out of the thallic membrane into the stroma and eventually out of the chloroplast cell into the atmosphere. Is this proton gradient? We saw something about some downhill movement in the electron chain. Electron carrier chain has higher affinity for electrons. As the electrons flow downhill through ETC, the energy they release is used to pump protons into the lumen. This builds a proton gradient, high inside the lumen, low in stroma. Okay, then let's continue. That was a longer break than expected, but let's continue. Of cyclic and non-cyclic phosphorylation. The subtopic under electron transport chain. Second subtopic, cyclic and non-cyclic photophosphorylation. Living organisms have the capability of extracting energy from oxidizable substances that store this in the form of bond energy. Special substances like ATP carry this energy in their chemical bonds. As I said prior to this discussion, like ATP, NADPH are molecules that act as intermediate. They have energy stored in their chemical bonds. It's a temporary storage of energy. And these are oxidizable substances, store energy in the form in bond energy in the form of bond energy. The process through which ATP is synthesized by cells in mitochondria and chloroplast is named as phosphorylation. What is phosphorylation? The synthesis of ATP. The synthesis of ATP through a synthesis of ATP in a cell is called as phosphorylation. If it is a synthesis of ATP, it's called phosphorylation. Photophosphorylation is synthesis of ATP from ADP and inorganic phosphate in the presence of light. When two photophosphorylation, as the name suggests, synthesis of ATP is phosphorylation. Photophosphorylation is the synthesis of ATP in the presence of sunlight, and inorganic phosphate is called as photophosphorylation. When the two photosystem work in a series, first PS2 and then PS1, a process called non-cyclic photophosphorylation occurs. The two photosystems are connected through an electron transport system as an earlier in the Z scheme. Both ATP and NADPH are synthesized by this kind of electron flow. Okay, we saw what phosphorylation is. Phosphorylation is the formation of ATP is called phosphorylation. Photophosphorylation, formation of ATP in the presence of inorganic phosphate and sunlight, is called as photophosphorylation. When the two photosystem work in a series, like in the Z scheme we saw prior, that this one, the Z scheme is here. In this scheme, if we see, this whole scheme is called a Z scheme, and this process which we saw where the sunlight will fall on the LHC, it will go to the secondary acceptor of the photosystem 2. It will get transported to electrons through the electron transport system, which will indirectly help in the formation of ATP, and these electrons are then again transported to LHC. They are not lost in the formation of ADP, like in the formation of ATP. They will be transported to the LHC under the photosystem 1, which will send electrons to the primary acceptor, and this will also help in formation of NADPH or reduction of NADPH. And this kind of phosphorylation is called as, this is formation of ATP. Clearly, if we see, this is called as a non-cyclic phosphorylation, where both the photosystems are involved. When both the photosystems are involved, it's called as a non-cyclic phosphorylation. Non-cyclic photophosphorylation occurs. The two photosystems are connected through an electron system as seen earlier in the Z scheme. Both ATP and NADPH plus H plus are synthesized by this kind of electron flow. When only PS1 is functional, the electrons circulated within the photosystem and the phosphorylation occurs due to the cyclic flow of electrons. A possible location where this could happen is in the stroma lamellae, while the membrane or lamellae of the grana have both PS1 and PS2. The stroma lamellae membrane lack PS2 as well as NADP reductase enzyme. The excited electron do not pass through NADP plus but is cycled back to the PS1 complex. through the electron transport chain. The cyclic flow hence results only in the synthesis of ATP but not NADPH plus H plus. Cyclic photophosphorylation also occurs and only light of wavelengths beyond 680 nm are available for excitation. Photosystem 1, this is a LHC, chlorophyll P700. Light will come here, electron will go, electron transport system, and come back here. This is a cyclic photophosphorylation. Before understanding this, we have to understand the locations of the systems, that is the photosystems. The photosystems locations of photosystem. PS2 first. PS2 present inside the lumen, inside the thylakoid membrane, present inside the thylakoid membrane. Also on the membrane, PS1, not PlayStation 1 but photosystem 1, present on the stromal lamellae, the connections we saw before. Present on the stromal lamella or lamellae, comma, it's also present outer membrane of thylakoid. Both PS1 and PS2 are present on the thylakoid membrane. This is the locations of photosystems where PS2 presents inside the thylakoid membrane, also on the membrane. PS1 present on the stromal lamellae, also on the outer membrane of thylakoid. Both PS1 and PS2 are present on the thylakoid membrane. That's for the locations. Now the process of the non-cyclic photophosphorylation is basically the Z scheme, which is this one. So now let's start with non-photophosphorylation. I mean cyclic photophosphorylation. Cyclic photophosphorylation. This happens at stroma lamellae, where only PS1 is present. Occurs mostly at times when light in the above the 680 nanometer range is present in excess, we can say, produces only ATP and not NADPH. It lacks, or stromal lamellae lacks, NADPH reductase enzyme. Very, very important, remember. Only the mechanism is generally not asked, only the specific enzymes are asked, and which, where this NADPH enzyme is lacking is also asked. Now that's with the cyclic photophosphorylation, and that's the main important points. Now the procedure, let's go with green. Go green. That's the sunlight effect. And the main process occurs here. It's not enough space to denote it here. ADP plus inorganic phosphate gives ATP. Electrons get recycled, hence cyclic phosphorylation. And I forgot to write the definition of phosphorylation itself. Phosphorylation, the synthesis of ATP, is known as phosphorylation. Think that's pretty much it about this one. Let's read it again so we can see if I missed anything. ATP synthesized in mitochondria and chloroplast is known as phosphorylation. Photophosphorylation is the synthesis of ATP from ADP and inorganic phosphate in the presence of light. When the two photosystems work in a series, first PS2 and PS1, a process called a non-cyclic photophosphorylation occurs. This is also a very common, understandable point, but very, very important to remember. The most possibility question: first PS2 and PS1 working together, that is called as a photophosphorylation. If only PS1 works, it's called phosphorylation. Let's write it down. If PS1 and PS2 work together, it's called a non-cyclic phosphorylation. Here NADPH also produced. Z scheme. If only PS1 is involved, it is called cyclic phosphorylation, photophosphorylation. But I am only writing phosphorylation here. I think that's all. If not, I'll write it again. The two photosystems are connected through the electron chain as seen earlier in the Z scheme. Both ATP and NADPH plus H plus are synthesized by this kind of electron flow. When only PS1 is functional, the electron is circulated within the photosystem and the phosphorylation occurs to cyclic flow of electrons. Possible location where this could happen in the stroma lamellae. While the membrane of lamellae of grana have both PS1 and PS2, the stroma lamellae membranes lack PS2 as well as NADP reductase enzyme. It's not NADPH but NADP reductase enzyme. NADP reductase enzyme. The excited electron does not pass on the NADP plus but is cycled back to PS1 complex through the electron transport chain. The cyclic flow hence results only in the synthesis of ATP but not of NADPH plus H plus. Cyclic photophosphorylation also occurs when only light wavelengths beyond 680 nanometer are available for excitation. Okay, that is the second subtopic. Now let's come to the third subtopic, that is chemiosmotic hypothesis. Let us now try and understand how actually ATP is synthesized in the chloroplast. The name is good. It's good to remember the name of this subtopic: chemiosmotic hypothesis. It's a kind of a theory. It might or might not be proven, but according to the textbook here, it is called as a hypothesis. It is a chemiosmotic hypothesis based on chemical osmotic pressure. Based on the name, we can understand it depends on osmotic pressure or something like that. Not osmotic pressure, like similar to osmosis. It's not directly related, but just similar. Let's see how so. Let us try and understand how actually ATP is synthesized in the chloroplast. The chemiosmotic hypothesis has been put forward to explain the mechanism. Like in respiration, in photosynthesis too, ATP synthesis is linked to development of a proton gradient across the membrane. This time there are the members of thylakoid. There is one difference though. Here the proton accumulation is towards the inside of the membrane, that is in the lumen. In respiration, protons accumulate in the intermediate space of the mitochondria where electrons move through the ETS. I don't know why they are mentioning chapter 4 in chapter 13, but we will, when I do the chapter, I'll explain that as well. Let us understand what causes the proton gradient across the membrane. We need to consider again the process that takes place during the activation of electrons and their transport to determine the steps that cause a proton gradient to develop. Okay. First thing, what is this chemiosmotic hypothesis? This theory has been put forward to explain the mechanism of ATP synthesis. To like explain how ATP synthesis happen, this theory has been put down, basically written or researched. If we take a respiration example, in respiration, photosynthesis also ATP is linked to development of a proton gradient across the membrane. Fine, I'll explain what happens in respiration a little bit. If we already know that mitochondria is the powerhouse of a cell, very basic knowledge, very much known. Mitochondria in a cell is the powerhouse of a cell, and ATP is synthesized in the mitochondria. For respiration purpose, ATP is synthesized in the mitochondria. So if we take this as a mitochondria, we know that, as I explained before, mitochondria has double layers. Mitochondria, chloroplast, nucleus are double layered cell organelles. They have two layers. Now, in mitochondria, there will be pumps on the membrane, proton pumps. It will be much more detail in the next chapter. Basically, due to these pumps, protons will get accumulated in this space. This is the interstitial space or the interboundary space or intermembranal space of the mitochondria. Protons will get filled here, and there will be a structure, that is the ATP synthase. No, I'm not sure. There will be a structure, basically all these protons which has abundant ADP molecules and inorganic phosphate. All the protons, because it's very concentration proton gradient, all the protons, because there is a kind of a efflux or basically a tap, all the electrons will try to flow, protons will try to flow out of this tap here, and ADP and inorganic phosphate are present here. ADP plus inorganic phosphate are present here, due to which when protons are passing here, ATP is produced. This is what happens in respiration. Much more detail in next chapter. There is one difference though. Here the proton accumulation, like what we are doing, we have already discussed this in the prior topic where like they asked where the PS2 is located, where the protons will be deposited. I think we didn't discuss it. I saw it. Anyways, yeah. In respiration, photosynthesis to ATP synthesis is linked or based on development of proton gradient across a membrane. ATP synthesis depends on formation of a proton gradient across a membrane. This time these membranes are of thylakoid. There it is mitochondria. In case of photosynthesis, for ATP synthesis, in photosynthesis case, we use thylakoid membrane and we create proton gradient between the thylakoid inner membrane, the lumen of the thylakoid, and the stroma. Stroma is the outer cytoplasm of the chloroplast, and lumen is basically the inner empty space of the thylakoid. Now, when we fill protons inside the lumen or the empty space of the thylakoid and get them concentrated there or accumulated there, this is what we call a proton gradient. Protons accumulate in the inner membrane space of the mitochondria when electrons move through the ATS. Okay, wait. Inside the membrane. Proton accumulations toward the inside of the membrane, that is the lumen, as I said. In respiration, protons accumulate in the intermembranous space here like this of the mitochondria when electrons move through the ATS. Okay, that's the end of the respiration reference, I think. Let us understand what causes the proton gradient across the membrane. We need to consider the process that take place during the activation of electrons and their transport to determine the steps that cause a proton gradient to develop. As I said before, the protons will get accumulated inside the thylakoid, that is in the lumen of the thylakoid. That is what is causing a proton gradient. Now let's see how this proton gradient is formed. This Since splitting of water molecules takes place in the inner side of the membrane, the protons or the hydrogen ion that are produced by the splitting of the water accumulate within the lumen of the thylakoids. That is the first method. That is what creates the, okay, I'll explain it later again. As electrons move through the photosystem, protons are transported across the membrane. This happens because the primary acceptor of electrons, which is located towards the outer side of the membrane, transfers its electrons not to electron carrier but to a hydrogen carrier. Hence, this molecule removes a proton from the stroma while transporting an electron. When this molecule passes on its electron to the electron carrier on the inner side of the membrane, a proton is released into the inner side of the lumen side of the membrane. Okay. The NADP reductase enzyme is located on the stroma side of the membrane along with the electrons that come from the acceptor of electron of PS1. Protons are necessary for reduction of NADP plus to NADPH plus H plus. These protons are also removed from the stroma. Hence, within the chloroplast, protons in the stroma decrease in number, while the lumen there is accumulation of proton. This creates a proton gradient across the thylakoid membrane as well as a measurable decrease in pH in the lumen. Okay, let's see the first method. Since splitting of water molecules takes place on the inner side of the membrane, the protons or hydrogen atoms that are produced by the splitting of water accumulate within the lumen of the thylakoids. We know that PS2 is located inside the membrane. Only PS2 is located inside the membrane of the thylakoid. Both PS2 and PS1 are present in the outer membrane of the thylakoid, and only PS1 is located in the stroma lamella. So, when splitting of water happens inside the lumen of the thylakoid, H2O splits, we will get 4 H plus. These H plus are also called hydrogen ions, or basically they are just protons. Single proton, nothing else. So they are protons. They get accumulated in the lumen where they are released, where they are split. There these hydrogen atoms, I mean protons, will get accumulate within the lumen of the thylakoids. As more protons are getting accumulated inside the lumen, more the proton gradient created. So that is the first method. Now the second method. As the electrons move through the photosynthesis, protons are transported across the membrane. As electrons move through the photosystem, we know that electrons move through, these are excited electrons, where photosynthesis, like these are excited electrons where the reaction centers produce these electrons and they are carried through the electron transport systems through the primary acceptor, secondary acceptors. And as they move through the photosystems, like they are moving from one system to second photosystem that we saw in the Z scheme, as they are moving, protons are transported across the membrane. Protons are transported across the membrane. This happens because the primary acceptor of electron, which is located towards the outer side of the membrane, transfers its electron not to an electron carrier but an hydrogen carrier. Hence, this molecule removes a proton from the stroma while transporting an electron, which this molecule passes on its electron to the electron carrier on the inner side of the membrane. Proton is released in the inner side of the lumen. Let's get this doubt clarified. After photosystem 2 absorbs light and excites p680 electrons are transferred to the primary acceptor. PQ is carrier located in the thylakoid membrane. It is special because it can carry both electrons and protons. That's why it's called a hydrogen carrier or the proton carrier. Let's see what they said. Electrons through the photosystem, protons are transported across the membrane. The primary acceptor of electrons, which is located toward the outer side of the membrane, transfers its electron not to an electron carrier but to a hydrogen or proton carrier. Hence, this molecule removes a proton from the stroma while transporting an electron. When this molecule passes on its electron to the electron carrier on the inner side of the membrane, electron is released into the inner side of the lumen side. This hydrogen carrier picks up two electrons from the primary acceptor of PS2. To balance charge, it takes up two proton H plus from the stroma. Okay, first let's understand the first line. I'm not sure why the PS2 absorbs light and excites P680, but these electrons should go to the acceptor, but I'm not sure why it's going to the primary acceptor. Then this PQ plastoquinone is a electron as well as a hydrogen carrier or proton carrier. When it carries two electrons or two electrons, it carries two protons. If it carries one electron, it carries one proton with it to balance the charge. And it is located in the thylakoid membrane. It's located in the thylakoid membrane. Electron pickup, outer stromocyte. PQ picks up this plastoquinone picks up two electrons from the acceptor. Okay, the second part of the process goes something like this. Basically, this is the PS2. PS2, the electrons will get excited from the reaction P680 reaction center, and these electrons will go to pheophytin. It's the primary acceptor of the PS2, primary acceptor of the photosystem 2. This primary acceptor will accept the two electrons and send it to PQ, which is basically, I think, plastoquinone or something. Plastoquinone, yeah, plastoquinone. This plastoquinone, it to balance the electron charge, it will also accept two protons, that is H, and it will become PQH2, which is soluble in the membrane of the thylakoid. And this is the electron chain. Basically, this is the electron transport chain. PS2, the primary acceptor, PQ, cytochrome B6F, plastocyanin, PS1, ferredoxin, and NADP plus. This is basically the Z scheme, but like a little more names involved. That's all. And as it goes from, as this PQ becomes PQH2, as it acts also as a H carrier, that is a proton carrier, it will become dissolvable in the membrane. This cytochrome, this is the next part of the electron chain, is actually present. It is embedded in the thylakoid membrane. This cytochrome B6F is embedded in the thylakoid membrane. So the next part of the chain, we only know PQ is the only H plus and electron carrier. Only this PQ acts as a H carrier. This cytochrome does not act as a H carrier. Meaning, although the electrons get transported to this cytochrome 6bf, and then to this, and then reduce NADV plus, that does not happen to protons. PQ is the only H carrier. These guys don't act as H carrier, that is proton carrier. So PQ will dissolve like in the membrane, and protons will get into the lumen, where because they get dissolved through the membrane, protons will go inside, increasing the proton gradient, whereas the electrons will continue to go through the electron chain. This is the process, as this is the second process to increase the proton gradient. The first was the water splitting inside the lumen, basically that one, and this one. This is the second step. Now let's see the third step. The NADP reductase enzyme is located on the stroma side of the membrane. Stroma side of the membrane on the thylakoid membrane itself. This NADP and reductase enzyme is located on the stroma side of the membrane, along with electrons that come from the acceptor of electrons of PS1. Protons are necessary for reduction of NADP plus to NADPH plus H plus. And although this third process doesn't directly remove electrons from the deposit electrons in the lumen, this process will reduce the amount of electrons in the stroma. It does not deposit them in lumen, but it reduces the number of protons. Basically, it will accept an electron, release one more. It will not release them, but like it will reduce one electron. So that's that. Hence, within the chloroplast, protons in the stroma decrease in number, while in the lumen there is accumulation of protons. This creates a proton gradient across the thylakoid membrane, as well as a measurable decrease in pH in the lumen. Why are we so interested in the proton gradient? This gradient is important because it is a breakdown of this gradient that leads to the synthesis of ATP. The gradient is broken down due to movement of proton across the membrane to the stroma through the transmembrane channel of the CF0 of the ATP synthase. The ATP synthase enzyme consists of two parts. One called the CF0 is embedded in the thylakoid membrane and forms a transmembrane channel that carries out facilitated diffusion of protons across the membrane. The other portion is called CF1 and protrudes on the outer surface of the thylakoid membrane on the side that faces the stroma. The breakdown of the gradient provides enough energy to cause a conformational change in CF1 particle of the ATP synthase, which makes the enzyme synthesize several molecules of energy packed ATP. Chemo Chemiosmosis requires a membrane, a proton pump, a proton gradient, and the ATP synthase. Energy is used to pump protons across a membrane to create a gradient or high concentration of proton within the thylakoid lumen. ATP synthase has channel that allows diffusion of proton back across the membrane. This releases enough energy to activate ATP synthase enzyme that catalyzes formation of ATP. Along with NADPH produced by the movement of electrons, the ATP will be used immediately in the biosynthetic reaction taking place in the stroma responsible for fixing CO2 and synthesis of sugars. What chemiosmosis requires is a proton pump, a proton gradient, and an ATP synthase. Energy is used to pump protons across the membrane. This raises enough energy to activate ATP synthesis enzyme and catalyze the formation of ATP. And as for how the ATP is formed above statement, the gradient is broken down due to the movement of proton across the membrane of stroma through the transmembrane channel of the CF0 of the ATP synthase. The ATP synthase enzyme consists of two parts, one called CF0 embedded in the thylakoid membrane and forms a transmembrane channel that carries out facilitated diffusion of protons across the membrane. The other portion is called CF1 and protrudes on the outer surface of the thylakoid membrane on the side faces of stroma. The breakdown of gradient provides enough energy to cause in the CF1 particle on the ATP synthase, which makes the enzyme synthesis several molecules of energy. Okay, basically, okay, we understood the how the protein gradient is accumulated. Now, if we see, let's assume this is a thylakoid. This is a thylakoid. There is proton gradient achieved. When proton gradient is achieved, there is no place for protons to go out, but there will be a special ATP synthase enzyme. This ATP synthase enzyme has two parts. It's like a tap. It has two parts, that is CF0 and CF1. CF0 and CF1. CF0 is a transmembrane channel. This channel connects both the inside of the lumen to the outside of the stroma. And because of this channel, not protons, which are concentrated, accumulated inside inside the lumen, have a way to come out. And this is through the CF0. There is a transmembrane channel. So obviously the electrons will get moving through here and they will come out of here, and which this energy in the this potential energy in the hydrogens, protons, that is repulsive force, plus plus repel, this force, this potential energy is enough for the formation of ATP. ATP is thus formed as the breakdown of this protein gradient is happening. More and more number of H plus come out of this ATP synthase enzyme consisting of CF0 and CF1, and more number of ATP molecules are produced. This is the ATP production chemiosmotic hypothesis. Now, as this ATP is produced, it is immediately used up for like the not dark reaction, the light reaction of production of glucose as sugar, basically synthesis of sugar. We know that this ATP is in the stroma, and in stroma, due to an enzymatic reaction, there will be production of sugar. This sugar is later converted into starch. That is called dark reaction, but is not, but that is not mentioned right now. ATP will be immediately used in the biosynthetic reaction taking place in the stroma, responsible for fixing CO2 and synthesis of sugars. Sugar is therefore synthesized. That's about this one. Now if I had to rewrite it, well, let's see the diagram. ATP synthesis through chemiosmosis. They did mention about the cytochrome B6F. This diagram is also very important, very important to remember. Let's start with PS2. Light falls on PS2. Now P680, PS2, this will send out electrons. Oxygen will come out. Okay, this is the whole, this is actually a thylakoid. This is a thylakoid membrane. This whole thing is a thylakoid membrane, a single thylakoid. On this thylakoid are embedded are the PS2, the photosystem 2. Photosystem 2 is embedded on the thylakoid. As it is embedded on the thylakoid, mention of plastoquinone is also here. Basically, the first process is splitting of water, where water is split and formation of H plus here. And more the second process, that is electron transport. Electrons are transported to plastoquinone, where the electrons, I mean the protons, are pulled from outside here, and as it is transported to the second chain, this will be, this accumulated H plus will get accumulated inside the lumen of the thylakoid. Then the electrons will again be transported to plastocyanin, the step of the electron chain. Then to P700, that is a PS1. This will help in NAD plus to H plus. This will basically, NAD plus will accept electrons and a H plus from the stroma, reducing the amount of H plus in the stroma to form NADPH. Thus, accumulation of H plus happens inside the lumen of the thylakoid. Now, this is the ATP synthesis. This is the CF0, the transmembrane channel, and this is the ATP synthesis CF1, which there is presence of ADP and inorganic phosphate here. As the H plus moves out, ADP will become ATP. Thus, ATP is synthesized. It is very annoying to write this again, but Okay, I'm not going to write it right now. Where are ATP? Next topic. This is not a subtopic, but like our next topic. Where are ATP and NADPH used? We learned that the products of right reaction are ATP, NADPH, and O2. Of these, O2 diffuses out of the chloroplast, while ATP and NADPH are used to drive the process leading to synthesis of food. More accurately, sugars. This is the biosynthetic phase of photosynthesis. This process does not directly depend. Okay, let's just write that one. Chemiosmotic hypothesis. Proton gradient between stroma and lumen, breaking of the gradient, ATP formed. This is the basic idea. Proton gradient. Protons getting accumulated in the lumen of thylakoid is called proton gradient. It is achieved via three ways: splitting of H2O inside lumen releases protons. The electron produced during the splitting of water Thereby it reduces the number of protons in the stroma. These are the three methods of creating a proton gradient. Now coming to formation of ATP synthesis. Formation of ATP. The electrons not electrons, protons inside the lumen pass through when a certain proton gradient is achieved. The protons inside the lumen will pass through the ATP synthase to out of the stroma, which results in the formation of ATP. And about ATP synthase molecule, there are two parts. Consists of two parts. CF0, a transmembrane channel, allowing passage of protons, and CF1, an opening leading to stroma and produces ATP as protons pass through it. That is ATP synthesis. ATP after formation is quickly used up to in the stroma for the synthesis of sugar, etc. I think that's all the information. The ATP synthase enzyme. The ATP synthase enzyme consists of two parts, one called the CF0, embedded in the thylakoid membrane and forms a transmembrane channel that carries a precipitated diffusion of protons across the membrane. The other portion called the CF1 and protrudes on the outer surface of the thylakoid membrane on the side that faces the stroma. The breakdown of the gradient provides enough energy to cause a conformational change in the CF1 particle of the ATP synthase, which makes the enzyme synthesize several molecules of energy packed ATP. That's pretty much it seems. Then let's start with the second topic. Where are the ATP and NADPH used? We learned that products of light reactions are ATP, NADPH, and O2. Of these, O2 diffuses out of the chloroplast, while ATP and NADPH are used to drive the process leading to synthesis of food, more accurately sugar. This is the biosynthetic phase of photosynthesis. This process does not directly depend on the presence of light, but is dependent on the products of the light, that is ATP and NADPH. Besides CO2 and H2O, you may wonder how this could be verified. It is simple. Immediately after light becomes available, the biosynthetic phase continues for some time and then stops. If then light is made available, the synthesis starts again. Okay, what they are saying is this is the biosynthetic phase. Biosynthetic phase is nothing more than the formation of sugar. The formation of sugar is, or formation of food is known as biosynthetic phase. In this phase, it does not directly depend on light like ATP and NADPH. It depends on the products of light that are ATP and NADPH. As long as there are presence of NADPH and ATP, this process, the biosynthetic phase, will continue. But if they stop, then this will also stop. But CO2 and H2O are still needed. If we want to verify how the biosynthetic phase depends on the products of light reaction, that is NADPH and ATP, we can simply expose the light, expose the plant to light. Biosynthetic phase will happen, food will be formed. But if we do not expose it to light and we keep it in dark for some time, the biosynthetic phase stops. So we can understand that it is a light dependent reaction, but light product dependent reaction. Can we hence say that calling the biosynthetic phase as dark reaction is a misnomer? Discuss this amongst yourself. Let us now see how the ATP and NADPH are used in the biosynthetic phase. Okay, we are going to see how the ATP and NADPH are used in the biosynthetic phase. We saw earlier that CO2 is combined with H2O to produce CH2ON, that is the empirical formula of sugars. It was of interest to scientists to find out how the reaction proceeded, or rather, what was the first product formed when CO2 is taken in that reaction are fixed. Okay, we are now going to see how this NADPH and ATP, we saw that like after ATP is produced, it's completely used up to produce sugar. That's the conclusion we came after our previous chemi-osmotics hypothesis in that. Now let's see how it actually gets used up. The scientists were trying to find out what is the first product formed after in this certain phase. Like how does, it's not like after hydrogen gets like water gets electrolyzed or split. It's not like directly just hydrogen fuses with the carbon dioxide, it forms C6H6. That's not possible. So there must be many intermediates between the formation of the glucose and reduction of carbon dioxide. So the scientists try to find that out, how this reaction proceeded, or rather what was the first product formed when the CO2 is taken into the reaction or is fixed. Just after World War II, after 1945, among the several efforts to put radioisotopes to beneficial use, the work of Melvin Calvin is exemplary. The use of radioactive 14C by him in algal photosynthesis studies led to discovery that the first CO2 fixation product was a 3-carbon organic acid. He also contributed to working out the complete biosynthetic pathway, hence it was called the Calvin cycle after him. The first product identified was 3-phosphoglyceric acid, or in short, PGA. How many carbons does it have? 3. Okay. So we saw until how scientists wanted to find out the first product. And just after the World War, where nuclear weapons we found out were radioactive, all based on radioactivity of metals, where nuclear fission happens, this releases massive amount of energy, this causes an explosion. That's how nuclear weapons work, where nuclear fission leads to massive amount of energy which causes an explosion. That is how a nuclear weapon works. And after World War II, there was some knowledge where after nuclear weapons were developed, there was some knowledge about radioisotopes being prevalent. Like, it was mostly known to the scientists about radioisotopes. And there are many experiments using radioisotopes. Using radioisotopes, many of the unknown experiments have been, like, many of the unknown, let's just say many of the discoveries were made using the radioisotopes, or can be proved using the had been proved using the radioisotopes. Among them, one is an experiment of Melvin Calvin. The radioactive isotope he used is very important to remember: C14. 14 is a mass number of the carbon, is a atomic mass unit of carbon. It has two extra neutrons compared to C12H6. This carbon atom has two extra neutrons, and it is used in an algal photosynthesis studies. And it's also a radioactive isotope. As for what an isotope is, isotopes are atoms having same atomic number but different mass number. The use of radioactive 14C by him in algal photosynthesis studies led the discovery of the first CO2 fixation product, that was a 3-carbon organic acid. Using this C14, he concluded that the first uh, product after of the photosynthesis was 3-carbon organic acid. And he not only gave the first product of the photosynthesis, but he also completed the whole pathway. Now, because he did that, it was named after him, called the Calvin cycle. Melvin Calvin cycle. It is called the Calvin cycle. This Calvin cycle is a pathway of production or synthesis of sugar. The first product identified was a 3-carbon organic acid. It was called the 3-phosphoglyceric acid, or it is in short called PGA. It has three carbon atoms. These products are very, very important to remember. This about the first three carbon organic acid, PGA, phosphoglyceric acid, 3-phosphoglyceric acid. Important to remember. The first product formed in the Calvin cycle. Let's see the whole Calvin cycle. It is given here, but it's after a few subtopics away. Scientists also tried to know whether all plants have PGA as the first product of CO2 fixation, or whether any other product was formed in other plants. Experiment conducted over a wide range of plants led to discovery of another group of plants where the first stable product of CO2 fixation was again an organic acid, but one with 4 carbon atoms in it. This acid was identified to be the oxaloacetic acid, or OAA. Since then, CO2 assimilation during photosynthesis was said to be of two main types. Those plants in which the first product of CO2 fixation is C3 acid, that is phosphoglyceric acid, that is the C3 pathway, and those of the first product was a C4 acid, OAA, oxaloacetic acid, that is a C4 pathway. These two groups of plants showed other associated characteristics that we will discuss later in this chapter. Okay, basically CO2 fixation is nothing but converting CO2 into sugar. Now the first product, with Melvin Calvin, he discovered the first product in some range of plants. It is PGA, phosphoglyceric acid. It was a 3 carbon acid, 3 carbon organic acid. Now scientists were curious to find out if all the plants in existence, their first product was phosphoglyceric acid itself. But they found out that that was not the case. The second type of plants they encountered were of C4 pathway. As the name says, this specific type of plants had their first stable product to not be a 3 carbon acid, but a 4 carbon organic acid called as oxaloacetic acid, represented by OAA. Since then, the CO2 fixation or the CO2 assimilation during photosynthesis was said to have two main pathways. That is the C3 pathway, where the first product, the C3 pathway, as we know, C3, 3 carbon atoms, C3 pathway, the first product formed is phosphoglyceric acid, and the C4 pathway, the first stable product formed was a 4 carbon organic acid, which is called the oxaloacetic acid. The two groups of plants showed other associated characteristics that we will be discussing later. That's about the two main pathways. The information given about the biosynthetic phase. Biosynthetic phase is the phase where production of sugar takes place. This process does not directly depend on light, but presence of ATP and NADPH. And then comes Melvin Calvin and the two pathways. Not much information, but still annoying to write. Biosynthetic phase. The production of sugar using ATP and NADPH, products of light reaction, is called biosynthetic phase. It happens when in light production of ATP and stops in dark. That is about the biosynthetic reaction. Melvin